So after we've gone through this chapter with electric potential, we now want to see how it can be useful once it's applied to life and medical physics, because that is what the course is about. So what we're going to cover are different topics from chemistry to molecular mapping and modeling to mapping the human body. So it's all about electric potential. And the first thing we want to talk about is just atomic interactions. So before going through that, we do have to understand the anatomy of an atom. If you do want to learn more about these types of interactions, you can always take physics 280 and 380, where we go in depth into each of these topics. So with the anatomy of an atom, I don't really have to explain this too much, but we know that atoms are made up of a positive nucleus and negative electrons, where the nucleus has protons and neutrons. So according to Coulomb's law, though, these two should attract, right? So you have the positively charged nucleus and you have your negative electrons. Based on Coulomb's law, you have a force that's going to want them to attract to each other. And even though the electrostatic attraction is what keeps them together, if you actually do the calculations, it would seemingly collapse because it would just be crushed within each other. So classical mechanics cannot explain this phenomena, but quantum mechanics can. So what happened was Niels Bohr identified or modified the Rutherford model by requiring the electrons to move in orbits of fixed size and energy. And radiation can only occur if the electrons jump from one orbit to another. So atoms are then considered stable within their own electron clouds representing their size. So what this means is that Coulomb's law does have limits once you go down to the nanoscale, but it doesn't mean we can't apply Coulomb's law to mimic the interactions between molecules. So for atomic interactions, we can look at atoms as point charges and as dipoles. So we're just going to ignore quantum mechanics and assume that the atoms don't overlap in space at all. So atomic distances are usually measured in angstroms. The smallest atom, hydrogen, has a diameter of approximately 1 angstrom. So here, what we're trying to model using electrostatic interactions is you have one electron here, another electron here, and they have a distance of r between each other. So you can potentially calculate the magnitude of electrical potential energy due to Coulomb interactions between two electrons by just saying U is equal to Q times V. I have KQQ over R, and then calculate this to be 14 electron volts. So this, to me, is considered a model. And in physics, especially in biophysics, life physics, or medical physics, it is important for us to create models for us to do calculations with, because there are so many parameters within the human body and within each of these systems that we do have to simplify things in order to create overviews and actually do calculations with it. So what types of electrical interactions are there? Just keep in mind, these are things that are just potential models for us to look at. You won't be tested on the actual mathematics of it, but because it'll be explained in detail in physics 280 and 380. So for now, just understand what types of interactions can take place. So we can have charge-charge interactions, charge-dipole, dipole-dipole, a dipole. You can have charge and induced dipole interactions, and you can have dispersion interactions. So let's just go through each of these visually so you can see what they represent. If we have a charge-to-charge interaction, it means that two molecules hold a charge, and we can just model them as charged, either a charged point charge or a charged object with a specific distribution of charge. So that means you have a little q and a big Q and a distance r in between, and that's simply Coulomb's law. Potential energy will decrease as the distance increases, proportional to 1 over r, based on V equals to KQ over r. If you have a charge and a dipole interaction, we now can consider one of the molecules a point charge and the other molecule a dipole. In that case, we actually have to consider what angle the dipole is at. And if we're just looking at one of these angles, then that's fine. We take a look at the charge-dipole interaction, and it's represented by the electric field generated is represented by this model. Next, if you have a dipole that rotates, then you have to sum up all of the possible different angles and configurations it has and come up with an energy that's averaged over all angles. If you have dipole-dipole interactions, this is like a snapshot. What if those dipoles do not move? This would be the equation for E as the electric field. Going forward, what happens if both of those do rotate? Then you have to sum up all of those different possibilities to get an averaged equation. Now we have a charge and an induced dipole force. This means you have a charge and you have a neutral object, but then that charge induces an uneven distribution of charge within that neutral object. So that means you have an induced dipole and a charge, and the electric field is then still going to be proportional to 1 over r to the 4. And last but not least, you have dipole-induced dipole force. So it's not when a charge induces a charge redistribution, it's when a dipole induces a charge redistribution, and that is going to be proportional to 1 over r to the 6. Now, the next thing to look at would be van der Waals forces. So van der Waals forces are considered weak and short-range electrostatic attractive forces between uncharged molecules. So they arise from the interaction of permanent or transient electric dipole moments. Just keep that in mind because this is like a group of different interactions that can take place represented by these. So we've already discussed that before, and they're just grouped under van der Waals forces. So the next application I want to talk about, apart from just applying potential to look at different electrostatic interactions, it's applying potential to create 3D electrostatic potential maps. So what this actually does is illustrate the charge distribution and charge-related properties of molecules in 3D. So having a knowledge of the charge distribution will help us determine how molecules interact with each other. So how do we collect data? We use what's called a molecular visualization program to display different styles of maps based on quantum chemical calculations. So here you have different charge distributions, and this is actually an equipotential map of how much potential you have as you get closer and closer to your charged object, H2O here, or farther away. And note that you do have a potential here, but also above. So this is how it's represented based on a electrostatic potential map, where you have the blue part at the bottom and the red part at the top. And this is a more general, it's a different style of map where green represents neutral in between the two charges. So the green would be here based on the 2D map. So these are just 3 different ways of representing the same thing, equipotential lines in 2D, 3D, and more of like a general 3D map of the whole thing. It's solid. So why are these maps useful? And where can we apply electrostatic potential maps in real life? So one good example is actually applying them so that you can take a look at the electrostatic interaction energies for specific molecules and proteins. So for example, in this picture here, you have the red area representing areas of active binding. So if there is an area of active binding, then the potential should be different because it is expecting to interact with a ligand so that it's, number one, it geometrically fits, and the electrostatic interactions are preferred for it to stay in that binding site. So here we have a lysozyme, and the negative region here is the binding site for a specific ligand. So what about the human body? We've so far looked at electrostatics and potential on the atomic and molecular level, but what about much larger objects? So now we're moving into medical diagnostics. Where can we find electric potential? We can find it when within our muscles, when they conduct electricity and generate force. Our brains are complex webs of neurons. Nerve cells are going to provide sensory input and motor control. And so a lot of medical technologies nowadays, they allow us to map the electrical activity of various organs of the body. The first one we want to go through is electroencephalography, or EEG. That's when you have this like cap on your head with a bunch of electrodes. And it's an electrophysiological technique that records the electrical activity of the brain. So based on what you're doing at a specific moment or what you're thinking about, different regions of your brain may fire a little bit more and have those neuronal signals activated. And you'll be able to see which lobe of the brain controls or becomes active based on certain movements. So the EEG is commonly used to diagnose epilepsy and other sleep disorders. So we'll take a look at these different types shortly. But how does it work and why does it work? Neurons communicate with each other through electrical events called action potentials, and they release chemical neurotransmitters from one neuron to another. So the process of firing an electrical signal will generate some sort of change in membrane potential, and that can be amplified and measured through EEG. So the mathematical theory behind EEG is a lot more complex than it looks. When the neurons are activated, local currents of electricity are produced. And so the electrodes on that EEG cap are going to measure these currents, and they use about 50 to 100 electrodes depending on which model you're using. There are two types of recordings that take place. There's a referential recording and there's a bipolar recording. It's important to know the difference between the two of them. A referential recording is going to give you the potential between each electrode and a fixed reference. So let's say you have the center of your cap. The referential recording will be the potential from one electrode to the center of the cap for each electrode. You then have bipolar recordings. Those will tell you the potential between an adjacent electrode to that electrode. So then you have different measurements of potentials to increase and improve your accuracy. The EEG voltage, VT, can be expressed by a physical waveform through Fourier series analysis. So you have what we call a time domain and a frequency domain. Through a time domain, that's what you would measure, then place it through a Fourier transform, which allows us to decompose an oscillatory signal into a bunch of sinusoidal signals like this. So then you can then get to a frequency domain and get an EEG voltage from that. The brain, however, isn't just a sine wave, and the waveforms are a lot more complex. So it does, it looks simple, but it's actually a lot more complicated, and we're not going to go through the math for that. So EEG instrumentation, you can see the cap itself has electrodes that are placed directly on the scalp. For a better signal, usually people do have to shave certain areas of their head so they get a better connection. There are different EEG wave patterns. This is something that you should know. So based on the wave patterns, they represent different brain states. So you should know that there's delta, theta, alpha, and beta. They're not really in any order. Just remember that beta is usually waking consciousness, where there's greater activity within your brain. So that's why there's a lot of spikes. Alpha is when you are relaxed, calm, and just not worried about anything. So in moments of no stress, you should be experiencing alpha waves of brain patterns where there's less spikes. Now you have theta, where you have level 1 and 2 sleep patterns, where there really isn't a lot of activity within your brain because you are in a resting mode. So either you are meditating or you are just about to fall asleep or just taking a quick nap. Now for the delta waves, that is when you have a deep dreamless sleep. It basically means that you have no activity or very minimal activity in your brain, and it is a very, very deep sleep. If you have REM sleep or if you have a very dream-like state within your sleep, then you will be able to notice changes within your sleep pattern or your brainwave pattern over here, because when we're dreaming, there is going to be brain activity. So just going back here, it will be important for you to know how to differentiate between these different patterns and also be able to match these patterns to examples of patients who have certain types of problems or disorders. So applications of EEG, tons of medical applications, helps you monitor brain activity, alertness, coma, brain death, etc. You should be able to tell from the brainwave patterns whether somebody, how alert somebody is. It's also good for locating areas of brain damage because one region can have very, very low brainwave patterns and another region can have very high brainwave patterns. So that might indicate an area of brain damage if one does not react the same as another. You can monitor cognitive engagement and reactions, and you can test the effects of drugs on brain activity. And most importantly, the EEG wave patterns are commonly used to investigate sleep disorders and cycles. That's probably where you've seen it used the most. And if you are interested in recent studies based on EEG, currently research, because it is already an established technique, recent research focuses on how to make the electrodes more comfortable and fit on the scalp without having to shave things off, while also improving the sensitivity to the brain's electrical activity. So right now it's more about optimizing the techniques and lowering the cost. Next, we have electrocardiography, EKG or ECG. The K actually comes from cardio from Greek, but electrocardiogram is spelled with C. And so it's a technique that's capable of not recording your brain's electrical activity, but your heart's electrical activity. Electrocardiograms are the graphs that are created from electrocardiography. So it's commonly used to A, check your heart's electrical activity, and B, diagnose diseases related to the cardiovascular system. Before we even talk about an EKG or ECG, let's take a look at the anatomy of the heart and see how an electrical signal is generated. So first, let's take a look at the SA node, or the sinoatrial node. What it's going to do is it's going to trigger an electrical impulse down here where the arrows are. It's going to cause the walls of the atria to contract. The stimulus is going to travel to the AV node, and then the stimulus is going to continue to the Purkinje fibers, and the ventricles are going to contract. The cycle is then going to repeat itself, so you have an electrical impulse that's transferred over to the ventricles to cause a contraction over and over again. So because of this electrical signal and this electrical activity, it is possible to try and record that signal and amplify it so that we can do measurements. So how does an ECG work? The technician is going to attach electrodes to the chest, arm, and legs. The electrodes are then going to be attached to leads and wires that are attached to the EKG machine. And from that, the heart rhythm is going to be recorded onto the machine and graphed onto an electrocardiogram. But how do we even measure the heart's electrical activity from the electrodes? It's fairly complicated in terms of the theory and the mathematics, but in general, it all has to do with trying to get more potential measurements from Einthoven's triangle. So Depolarization and repolarization of cardiac cells can be recorded from the surface of our skin through electrodes, just like we did with the EEG. So as the SA node triggers an electrical impulse, the action potential moves along the heart. One part of the cardiac tissue is depolarized, so I'm going to start here. This is the SA node, and we're going to follow the impulse down to the ventricles. So the cardiac tissue is depolarized, and another part is polarized, creating a dipole. So you have a negatively charged region and a positively charged region. The direction and magnitude of that dipole of the heart at any instant is called the electrical axis of the heart. So that net dipole I'm talking about is over here. And that vector is going to originate from Einthoven's triangle. So if you have 3 different electrodes over here that are taking measurements, you have negative to positive, and another negative to positive over here. These are the leads where you can represent the potential from one place, from one location to another. You combine these 3 vectors together and you get a net dipole that's called the electrical axis of the heart, so that you know what direction the electrical wavefront is going towards, that's created by the depolarization of the heart. So at least understand what Einthoven's triangle does for us, and why that electrical axis of the heart is important. But how does the recording actually work? It's all because of amplification of small potentials. So the working principle of an ECG is that you have many small potentials on the surface of your body from the heart, and so you have to amplify all of these potentials in order to generate some sort of signal. The electrodes have to be placed at well-defined anatomical positions on the body surface. So I'll explain this diagram in a bit. You can image the body as an equilateral triangle, which is Einthoven's triangle, and you can see 1, 2, 3 spots over here to create Einthoven's triangle based on where the heart is. Usually, there's a 12-lead ECG that uses 10 electrodes. You have bipolar limb leads. So you have 1, 2, and 3. So 1, 2, and 3 over here. You have unipolar augmented limb leads. So you have AVR, AVL, and AVF. So if you take a look, there's actually 2 triangles now. One from here, another one from here. And then you have unipolar chest precordial leads. So these are the red ones going across. So what this does is give you a bunch of different directions of potential that you can measure. And each lead is going to view the heart from a different angle to see if there's anything wrong with its signal propagation. So where can we apply ECG? Electrocardiography is a very commonly performed cardiology test. It's actually non-invasive and very fast and harmless. I'm pretty sure most of us have done one already. It's commonly used in medical diagnostics to measure the rate and rhythm of heartbeats, the effect of cardiac drugs, the function of electrical implants and pacemakers, and of course the presence of any heart abnormalities, and whether you have an enlarged heart chamber or not. So there are very many applications for ECGs, primarily in sports, primarily in people with heart diseases, or to check to see if you have a heart disease. And it's all because we are able to amplify the signal of potential received from different electrodes and leads on your body. Now let's go through a summary of electric potential. We're going to go through key concepts and mainly just important equations. So what did we go through? We defined work. We already know what the equation for work is. We defined the potential difference as the negative of work performed, which makes sense because the more work you put in, the less you're giving for potential energy. You have the change in potential energy being equal to this equation over here. But most importantly, we know delta V is equal to delta U over Q. That's the electric potential difference. And if we want to figure out the electric potential difference from point A to B for a continuous charge distribution, we use this form. And this is the most common form for delta VAB. Next, we have a point charge potential represented by KQ over R. But once again, if you have a continuous charge distribution, you're going to want to use this form of potential, not potential difference, but just potential. For discrete charges, you would use this. The last equation we went through was another definition of the electric field and how we can calculate it using potential. And that's if you use a potential gradient, you will be able to calculate the electric field by knowing its X, Y, and Z component based off of the gradient of potential. So next, we're going to be taking a look at conductors, capacitors, and how we can leverage all of the knowledge that we have learned for electric force, fields, and potential to analyze different situations and charge distributions.